Hello, hi, a very happy good evening. So this is Krishna Ma'am and we've been talking about the story of life on earth, and that's biology. So thank you so much for joining my highlight series. So today we are talking about respiratory pathways in detail. So we are going to highlight the important topics in this chapter, that is respiration in plants. The most difficult part, or the most Herculean part of this topic is your glycolysis, your Krebs cycle, you would say, your link reaction, fermentation and ETS. So we are going to highlight all these respiratory pathways today. So that is our agenda. So today it will be part 1, where I'll be talking about the first and the most important pathway, the primary pathway that happens in all living organisms, that is glycolysis. So then we'll further continue with your link reaction and fermentation. Tomorrow we'll talk about your Krebs cycle and your ETS. Alright, so that is the plan. So we will see from scratch why this has to happen and how it is happening. So usually students tell me, ma'am, glycolysis is very huge, you're not able to remember the enzymes. So come on, if you're not able to remember a couple of enzymes and this 10-step pathway, how are you going to do medicine? So that is the lifelong devotion. So I would say at least 10 years you have to devote your heart and soul to doing medicine, right? So only then you'll become a doctor, proper doctor, whom people or patients will trust, right? So let's see what we have to talk about in respiration and plants. So this is one of the highlight series. All right, yes, so let's continue. Before we continue, just give me a minute. Alright, so let's move forward. So respiration in plants, so actually it starts off with glycolysis. So this is the primary process that happens in all organisms, right? So there's a difference that happens in aerobic respiration and in anaerobic respiration. So as you know, aerobic is in the presence of oxygen and anaerobic is in the absence of oxygen, right? So when I say glycolysis, so glycolysis is common to all living creatures, be it a microorganism, be it a plant, be it an animal, be it a human. So everyone, the common process is glycolysis. Just like every photosynthetic plant undergoes C3 cycle, every living organism undergoes your glycolysis. So that is the primary process that happens in every living individual on the globe, right? Okay. So after that, the fate divides itself into two. One is aerobic respiration, the other is anaerobic respiration. So aerobic organisms, that is organisms which breathe in the presence of oxygen, after your glycolysis, they have what is called as the link reaction, which links your glycolysis and the third one, that is your Krebs cycle. So the one that forms a link between the glycolysis and the Krebs cycle is your link reaction, and it is a part of your aerobic respiration, and after your Krebs cycle you have your ETS happening. Alright. So in case of anaerobic organisms, where again glycolysis is common, after glycolysis you have the process of fermentation happening. There are two types of fermentation. One is alcoholic fermentation, the other one is lactic acid fermentation. But the entire question lies, or it can be simplified to one line. Why should all this happen? Why the hell all this is happening, right? So that is the simplest question that all of you have in mind. So just like we saw in photosynthesis, very categorically or in a very simplified way, how first light falls on the plants and how the chlorophylls make up the ATP, NADH and oxygen, and how this ATP and NADH are used in fixing the carbon dioxide and producing glucose, and how this glucose is being transported across the plant, right? So that was very simple, and that was very organized. Now, so when you see in photosynthesis, the products of light reaction are ATP, NADPH and oxygen, right? So this oxygen diffuses into your mitochondria. So when it diffuses into your mitochondria, what is happening? So actually, apart from photosynthesis, plants make photosynthesis to produce glucose, right? So in the process of making glucose, it actually utilizes ATP. But plants also need ATP for various other processes, right? So you eat to gain energy, not only for studying, but for various other activities. Your body needs energy to cleanse the cancerous cells, to cleanse the toxic cells, etc. Your body needs energy to repair the damage that's been happening internally, which you and I do not know, or which you and I cannot see, right? So much in the same way, so plant needs ATP for its own energy, or for its own day-to-day activities. So thus the ATP for those processes are produced by the process of respiration. Just like you eat, for example, say you had chapati and dal in the afternoon, so that chapati and dal breaks down to form the simplest molecules, be it amino acids, be it monosaccharides, be it fatty acids or glycerol, be it simplest glucose. So these molecules, the primary metabolites, are then further used to produce the other molecules, and in turn energy is stored in your body, right? So that is the main reason why respiration has to happen. So in respiration, the carbon-carbon double bonds are broken down to give you energy. The food that I eat is actually broken down. Say for example, I had a bowl of rice today, so the rice has starch, right? So the starch is broken down in my body as glucose, which is a monosaccharide, and now this glucose is used to give me more energy by moving into the process of Krebs cycle and in ETS. So this is the entire background story of why respiration is happening, right? So understood? So glycolysis is the common process, so it occurs in all living organisms irrespective of whether they are microbes, plants, animals or human beings or complex animals, right? So after glycolysis happens, which is common to both these kind of organisms or both these groups of organisms, right after glycolysis, in case of aerobic organisms, so there is a link reaction that is linking your process of glycolysis in the next process of Krebs cycle, which will be in turn followed by your electron transport system, right? So talking about your anaerobic organisms, so they do not have this link reaction in Krebs cycle, as you can see on the tabular column. So right after your glycolysis, so they undergo a process called as fermentation. So you would have heard about fermentation, so we have read about fermentation in biotechnology chapter. So that is what happens, all right? So let's move forward and look into what is this glycolysis all about. So today our focus remains only on glycolysis, the most fundamental process and the most common process to both these kind of organisms or to both these groups of organisms, all right? So this aerobic respiration, so glycolysis, link reaction and fermentation will be our agenda today, but let's see how much we can finish. So let's move forward. So respiration in plants, yeah, so it is divided like this, aerobic and anaerobic respiration. So I guess I briefed you the difference between both, so how it happens, what is common, so what happens later. So now moving further, so we come to this concept of glycolysis. Yeah, so it all starts with glycolysis, as the meme says. So we are going to begin from glycolysis. Okay. Hi W, yes, I'm very, very good. Thank you so much. I hope you're doing brilliant. Thank you once again for joining my session today. So please don't forget to like, share and subscribe. And I also hope your preparation is going very positively. And yes, you're revising and you're well prepared and you're ready to face the challenge of your NEET 2022. All right, so let's continue. Yes, so as the meme says, it all continues or it all starts with your glycolysis. So what is this glycolysis? So the word glycolysis is not of an Indian origin. So because Indian, the roots base goes around to Sanskrit, right? But this glycolysis term belongs to a Greek word where you can split glycolysis into two parts, glyco, then lysis. So glyco means sugar, lysis is breaking down. So you break down sugar to give you your pyruvate. All right, yes. So in the Greek word glycolysis, so I can split like this, glyco is sugar, lysis is splitting. I'm sorry, I've written glycolysis once again. Okay, yes, I'm suffering from fever plus headache plus vomiting for 1 week. Oh my God, so why did your health deteriorate? What is that you're doing? So come on guys, your health is of priority. So let your studies, everything else can wait, but not your health. So health is worth everything. So please take care of your health. So come back, gain your strength, gain your stamina. Only then you'll be able to study. All right. And at the same time, please make sure you take good care of your health, because that is what is your baseline. So when you fall sick, you will not be able to concentrate. As a result, you will not be able to do your best for the preparation. So please take care of your health. W, it's okay if you're not well. So please take 2-3 days more to gain your stamina, your energy, and then come back with full force. All right? Yes? Okay. So when we were talking about photosynthesis, what is the equation of photosynthesis? The Equation of photosynthesis is carbon dioxide plus water, which gives you C6H12O6 plus oxygen, right? So we know this oxygen moves into mitochondria, and this is the end product of photosynthesis, that is glucose, right? So this is the glucose that we are going to break down here, all right? So glycolysis, we are going to break the glucose molecules, right? Okay. So this pathway of glycolysis was designed by three eminent scientists. The name of them is Gustav Embden, Orton Meyerhoff, and J. Parnas. Okay, so these are the three scientists who together formulated your glycolysis process. Okay, so in short, it is also called as the EMP pathway. It is called as EMP because of this reason: Embden, Orton Meyerhoff, and J. Parnas pathway. All right, yes. So in anaerobic organism, glycolysis is the only major pathway that is happening, because after glycolysis it is only fermentation, right? So in case of anaerobic organisms, so this is the only major pathway that is happening. So where does it happen? So we are talking about the general location in both aerobic as well as anaerobic organism. Where does your glycolysis takes place? Your glycolysis takes place in the cytoplasm of the cell. So my abbreviation for CP, or my full form for CP, is cytoplasm. It happens in the cytoplasm of the cell. So what is the cytoplasm? So the contents inside a cell, so all the organelles are contained within the cytoplasm of the cell, or the space inside the cell is called as a cytoplasm. All right, yes. So here what is going to break down? So glucose. So when I say A is going to give, so when I write a chemical equation like this, A gives me B, so we call A to be the substrate, right? And we call B to be the product. So in glycolysis, the glucose molecule is going to break down into pyruvate molecules or pyruvic acid molecules. So thus my glucose is my substrate. It is a slightly different modification to it. It is called as respiratory substrate because the process of respiration is happening. Okay, so this is your respiratory substrate. So what is your respiratory substrate? That is glucose, right? So what is the end product that you're going to get? You're going to get pyruvate or pyruvic acid, right? Okay, so what is happening in the process of glycolysis is that the glucose is oxidized partially to give you pyruvic acid. So we start the process with glucose and we end it with pyruvic acid. So in between it is getting oxidized. Clear? Yes? So where does this glucose come from? So just now in the previous slide I told you, so this glucose comes from the end product of photosynthesis, but it is actually as sucrose. So when your plant makes glucose by the end product of photosynthesis, it is quickly converted into sucrose, and in the form of sucrose it is transported through the phloem. So if you have studied phloem transport in plants, it is transported in the form of, the food is transported in the form of sucrose, right? So when it has to undergo the respiratory pathway, the sucrose is broken down into glucose plus fructose, and this glucose and fructose enters your glycolytic cycle. Fine? Yes? So your sucrose is broken down into glucose plus fructose, because probably when I get a 20,000 cash, I will make sure I have only 1,000 rupees and the rest everything I'll put it in the bank so that I can use GPay and PhonePe, right? So much in the same way, when the plant produces glucose, it will see that I have to transport it, I have to keep it safe, so I'll convert it quickly into sucrose and I'll transport it and keep it safe. When there's a need for respiration, so that sucrose will be broken down into glucose and fructose in the presence of an enzyme that is called as invertase. So now this glucose and fructose will enter the glycolytic pathway. All right, yes? Okay, so what happens in glycolysis? So glycolysis is a 10-step process. We are going to see how this glucose is broken down into pyruvate, or how this glucose is partially oxidized into pyruvate. All right, yes? So here we go. So step 1. So the glucose is broken down into glucose 6-phosphate. So literally a phosphate group is getting added to the glucose molecule. So when a phosphate group is getting added to the glucose molecule, or any molecule, whenever a reaction has a phosphate group getting added, so that reaction is called as the phosphorylation reaction. So if you remember in mineral nutrition, when we are talking about the macronutrient phosphor, which is uptaken in the form of phosphoric acid, so it said the use of this mineral element or this macro element is phosphorylation. So phosphorylation means addition of phosphate. Okay, yes. So first step, glucose is getting an additional phosphate group at the 6th position. So that is why glucose to glucose 6-phosphate. So this happens in the presence of an enzyme, that is hexokinase. Whatever is written in between, that is your hexokinase. Sorry, that is your enzyme. Okay. So everything in red throughout the slide will be your enzyme. So the first step, glucose is getting converted into glucose 6-phosphate in the presence of hexokinase. Okay. But whenever phosphorylation is happening and whenever kinase group of enzymes are involved, there is an involvement or participation of ATP as well. So whenever phosphorylation reactions happen, what is phosphorylation reaction? Addition of phosphate. Okay, whenever phosphorylation reaction happens, kinase is involved. Here the kinase is hexokinase. At the same time, ATP also participates in this process. So here your ATP is needed and it is converted into ADP, meaning energy is supplied for this conversion of glucose to glucose 6-phosphate. Okay, right. So step number 2, so this was your step 1. So now we are in step number 2. So step number 2 says glucose 6-phosphate gets converted into fructose 6-phosphate. So glucose and fructose are isomers, right? So fructose is the sweetest sugar and glucose is the normal blood sugar, right? Yes. So now your glucose 6-phosphate is getting converted into fructose 6-phosphate. So glucose and fructose are isomers. So the enzyme is isomerase. So one term, phosphohexose isomerase. Both glucose and fructose are 6-carbon molecules, so it is hexose. Both of them have a phosphate group, so it is phosphohexose isomerase. What is step 1? Glucose is getting converted into glucose 6-phosphate, which is a part of your phosphorylation reaction. The enzyme is hexokinase. The second step is your glucose 6-phosphate is getting converted into fructose 6-phosphate, which is isomeric way, right? So glucose is an isomer, glucose and fructose are isomers, right? So here the enzyme is isomerase, but since both glucose and fructose are 6-carbon molecules, it is hexose. And since they have a phosphate group, it is phosphohexose isomerase. Clear? Yes? So this is step 2. Now we go to the next step. Fructose 6-phosphate becomes fructose 1,6-bisphosphate. So the full form is fructose 1,6-bisphosphate. So bis or bi means 2. So long they had only one phosphate at the 6th position, right? Now they are having one more phosphate at the 1st position as well as in the 6th position. So they are having phosphate at 2 positions. So that is why it is fructose 1,6-bisphosphate. Okay? Yes? Clear? Done. So once again here you need to give ATP because it is again phosphorylation reaction. If phosphorylation reaction is involved, kinase is involved, and if kinase is involved, ATP is involved. So ATP is utilized to convert your fructose 6-phosphate to fructose 1,6-bisphosphate. So the enzyme is kinase. It has a phosphate group, but who has the phosphate group? Fructose. So it is phosphofructokinase. First step is phosphorylation reaction. Third step is again phosphorylation reaction. So what happens? Glucose is getting converted into glucose 6-phosphate in the presence of hexokinase where ATP is utilized. In the second step, the glucose 6-phosphate is getting converted into fructose 6-phosphate in the presence of an isomerase enzyme, and since both glucose and fructose are 6-carbon molecule, it is hexose, and since both of them have a phosphate group, it is phosphohexose isomerase. So that is how you logically remember it. Alright, yes. So we usually have a notion biologists actually memorize things. No, we do have a logics. Otherwise, it's not possible to memorize everything. Biology is an ocean, right? Alright, so coming to step 3, so this fructose 6-phosphate gets another phosphate group added at the first position also. So it is fructose 1,6-bisphosphate, right? Again, it's a phosphorylation reaction, so the enzyme should be kinase, but this is only fructose on both the sides, so it is fructophosphose. Because they have a phosphate group, so it is phosphofructokinase. Clear? Yes? Fine. So now comes the next step, that is step 4. This fructose 1,6-bisphosphate is now getting converted into 2 products. What are the 2 products? DHAP and G3P. DHAP stands for dihydroxyacetone phosphate, and G3P stands for glyceraldehyde 3-phosphate. So now your fructose 1,6-bisphosphate is getting converted into 2 products, that is DHAP and G3P. DHAP is your dihydroxyacetone phosphate and your G3P is your glyceraldehyde 3-phosphate. Fine? Yes? Here the enzyme is aldolase. But let me tell you one more thing. This DHAP and your G3P are also isomers. They are also what? They are also isomers actually. So this DHAP can be reversibly converted into glyceraldehyde 3-phosphate, and this glyceraldehyde 3-phosphate can be reversibly converted into DHAP. Right? So now instead of considering both of them, I consider 2G3P. It is easier to understand. From the next step, I will be considering 2G3P. So the first 4 equations are clear? Yes. Any doubts so far? So what is the first step? Glucose to glucose 6-phosphate, then glucose 6-phosphate to fructose 6-phosphate, then fructose 6-phosphate to fructose 1,6-bisphosphate. So this fructose 1,6-bisphosphate gives 2 compounds, DHAP and glyceraldehyde 3-phosphate, which are in turn isomers. Clear? Okay, very good. So 4 steps are done. So we go to the next one. Now, why am I saying 2 molecules of G3P is because both DHAP and your glyceraldehyde 3-phosphate is going to have the same fate, right? So if you study from Living World or if you study from Chemical Coordination and Integration, you have to finish all the chapters too. It does not make any difference. What makes a difference is how you can understand. Right? 𝗏𝗵𝗃𝗧𝗏, yes, hello, welcome to my session. So thank you so much for joining me today. So please do like, share and subscribe. Okay, so that's why I'm considering DHAP as well as G3P because both are reversibly convertible, right? So I'm having 2 molecules of G3P considering the DHAP as well. Okay, so 2 molecules of glyceraldehyde 3-phosphate now gives me 2 molecules of 1,3-bisphosphoglycerate. So this is bisphosphoglycerate. So here I am having the phosphate group at the 3rd carbon, but here I am adding the phosphate group at the 1st position as well. Alright, so here the enzyme is glyceraldehyde phosphate dehydrogenase. So dehydrogenase is removal of carbon dioxide as well as sometimes phosphate gets added here. Alright, so what happens? Glyceraldehyde 3-phosphate changes its orientation to become 1,3-bisphosphoglycerate. Fine? Yes? Okay, so now what happens here? Since there is 2 molecules here as well as 2 molecules here, 2 molecules of NADP are broken down into 2 molecules of NADPH plus 2 molecules of H+. So here energy is gained, right? So far we have been using energy, but here it is energy is getting gained. Kinase or nase are same. See, NASC is the end for enzymes. Anything that is an enzyme ends in ASC. pectinase, ligase, polymerase, everything that ends in ASC is an enzyme. Okay, W? Yes. So here glyceraldehyde 3-phosphate, so fructose 6-phosphate and glucose 6-phosphate, it was very easy. But here glyceraldehyde 3-phosphate to 1,3-phosphoglycerate, the configuration changes, so the enzyme is dehydrogenase. Fine. So this is our step 5. So step 6, 2 molecules of 1,3-bisphosphoglycerate gets converted into 2 molecules of 3-phosphoglycerate. So 1 phosphate is getting removed, right? Yes. So what is getting 1,3-bisphosphoglycerate is getting converted into, 1 phosphate group is getting added here. So 1 and 3rd position has a phosphate group, that is why it is bisphosphoglycerate. Now it is 3-phosphoglycerate. There are 3 phosphates. Imagine that way. It is easier to correlate. So now when phosphorylation is happening, so here it is very simple, kinase is there. Whenever kinase is being used, but see on both the sides it's phosphoglyceraldehyde, so it's phosphoglycerate kinase. So whenever kinase is utilized, ATP is involved, but here ATP is produced. Since you have 2 molecules, here also 2 molecules of ATP are produced, right? No, W, it is not that way. It is in the way that I told you. So what happens? We have fructose 1,6-bisphosphate giving us DHAP as well as glyceraldehyde 3-phosphate, which are reversible. So we are considering 2 molecules of glyceraldehyde 3-phosphate. This changes its conformation to produce 1,3-bisphosphoglycerate. This 1,3-bisphosphoglycerate now gets converted into 3-phosphoglycerate. Now this 3-phosphoglycerate becomes 2-phosphoglycerate. It reduces 1 phosphate, right? So this is 3-phosphoglycerate and this becomes 2-phosphoglycerate in the sense that 1 phosphate is removed. So the enzyme is mutase. Look into both the sides. If this is glyceraldehyde 3-phosphate, this will be glyceraldehyde 3-phosphate dehydrogenase because the substrate is getting converted into the product. On both the sides it is glycerate, so it is phosphoglycerate kinase. So here it is phosphoglycerate, here also it is phosphoglycerate, so it is phosphoglycerate mutase. Alright, yes. So now these 2 molecules of 2-phosphoglycerate will give you 2 molecules of PEP. So PEP stands for phosphoenol pyruvate. So we have read this as the primary carbon dioxide acceptor in your C4 cycle, right? So here the enzyme is enolase. So this is step 5, step 6, step 7, step 8, right? The last step is when your PEP is getting converted into the final product, that is pyruvate. So here whenever pyruvate is again, so phosphoenol pyruvate to pyruvate, so again kinase is being used. Whenever kinase enzyme is involved, ATP is involved. So since there are 2 molecules, so again you have the production of 2 ATP molecules. So it is 2 ADP plus 2 PI, which gives you 2 ATP. This is your entire process of glycolysis. So what is it? Once again we'll revise quickly. So we'll revise this slide first and then we'll revise the entire thing. Okay. So first of all, what happens? We are considering 2 molecules of glyceraldehyde 3-phosphate, which becomes 1,3-bisphosphoglyceride, right? So now this becomes 3-phosphoglyceraldehyde and then 2-phosphoglyceraldehyde, then phosphoenol pyruvate, then pyruvate, right? So now are you able to remember the enzymes as well? Right? Ma'am, you said 10-step process, but you have only 9 steps. Let me tell you, in this previous slide, these 2 interconversion is considered as 1 step, which I haven't numbered. If you number it, it will become 10 steps in total. Alright? So 10 steps, 10 enzymes you have to remember, and I've given you clues how to remember the enzymes. Right? So first step and third step, ATP is utilized. okay? And where energy is produced? So energy is produced in step 5, step 6 and the last step. So energy is utilized in step 1, step 3, energy is produced in 5, 6 and the last step. Clear? Yes. And if you see, there are two equations where I have star marked. So this is one equation I have star marked, and if you see, this is one equation I have star marked. Why? What is the reason for star marking is that these reactions are irreversible. Wherever I haven't star marked, it is reversible. Wherever I have star marked, it is irreversible. There are only two irreversible reactions in your entire glycolysis, that is step 3 and the last step. Only two steps are star marked. Wherever I have star marked, iska matlab hai it is not reversible. It is irreversible steps. Fine? Clear? Yes? Okay. So now we are going to see how many energy is consumed and how much of energy is gained. So in step 1 and in step 3, ATP was utilized. What were the two steps? So first of all, your glucose was getting converted into glucose 6-phosphate. So for this, we had to supply ATP. This is step 1. Your third step was your fructose 6-phosphate. So your fructose was getting converted into fructose 1,6-bisphosphate, right? So again, for this ATP was utilized. So 2 molecules of ATP were utilized, were consumed for the reaction. Yes. So how many did we gain? So in step 5, we gained 2 molecules of NADH. So when we had 2 molecules of glyceraldehyde 3-phosphate, which in turn gave us 2 molecules of 1,3-bisphosphoglyceride. So we had 2 molecules of NADPH getting produced, right? So immediately right after that, we had another step where we had 2 molecules of 1,3-bisphosphoglycerate giving us 2 molecules of 3-phosphoglycerate. So this step also gave us 2 molecules of ATP. And then the last step, that is the 9th step. So 2 molecules of phosphoenol pyruvate gave us 2 molecules of pyruvate or pyruvic acid. So again 2 ATP was gained. So this is the ATP that was consumed and this is the ATP that is gained. Right? Yes? So now let's calculate. So now let's do the calculation. So we have 2 ATP, 2 ATP. So totally 4 ATP we are getting from the glycolysis process and we are getting 2 NADH. Sahi to hai? That's what I have written. 4 ATP and 2 NADH is what we have gained. Right? So how much is consumed? Here 1, here 1. 2 ATP is consumed. So if I get 4 rupees as my profit and if I have invested 2 rupees, what is my ultimate profit? Only 2 rupees. Because first I have invested my 2 rupees, first I have to put that 2 rupees back in my account. And the profit that I have earned is 2 rupees, right? So here the net gain would be 4 minus 2. So the net gain of ATP would be 2 ATP plus 2 NADH. So what it will be? 2 ATP plus 2 NADH. So this is the net gain of energy from glycolysis. You can convert NADH also in terms of ATP. So 1 NADPH is equal to 3 ATP. Here I have 2 NADH. So how many ATP it will give me? 6 ATP. So 6 ATP plus 2 ATP, totally 8 ATP also you can say. When you are saying it in terms of both ATP and NADH, it is 2 ATP and 2 NADH. But if you're saying everything in terms of ATP, it is 8 ATP. Alright? Yes, W, absolutely. So glycolysis is clear now? No doubt? Pakka? So one huge topic is done. So you'll never forget glycolysis. Can you give me the surety? Okay. So now what happens after glycolysis is the fate of pyruvate. So the end product of your glycolysis is your pyruvic acid or your pyruvate. So now what happens? In case of aerobic respiration, this pyruvate enters inside your link reaction. Now the link reaction will be followed by Krebs cycle, and Krebs cycle will be followed by your electron transport system, right? In case of anaerobic organism, it can either undergo lactic acid fermentation or alcoholic fermentation. So your pyruvate can undergo fermentation. Say for example, it is undergoing lactic acid fermentation, it will form lactic acid. So this process can be seen in case of your mussels, in case of your lactic acid bacteria. In certain anaerobic organisms, your pyruvate can be converted into alcohol. This is your alcoholic fermentation. So this alcoholic fermentation can be seen in organisms like yeast. Fine. So with this, I have covered glycolysis as well as fermentation process. Done? Any doubts so far? So glycolysis and fermentation is covered. So tomorrow I will cover your link reaction, your Krebs cycle, and if possible your ETS. Okay? Yes? So far clear? Yeah. So far clear? Yes? Chalo. So we'll stop here for today because I guess I've taught you the one major important or the primary process that is needed in respiration. So I hope you enjoyed today's lecture. If you genuinely enjoyed, so please do like, share and subscribe. So please let me know what are your topics which are really tough for you or the topics that you're weak in. So I will do a highlight series. So I will highlight the important concepts so it will be easy for you. So I hope you really enjoyed. So I equally enjoyed teaching you. So thank you so much. So tomorrow at 3:15 I will continue with the respiratory pathways 2, wherein I'll talk about the Krebs cycle, the link reaction and the ETS. So thank you, and once again please don't forget to like, share and subscribe. So stay tuned, take care and bye.